One of the most intriguing and enduring questions in animal behavior is that of mate choice. Why do females choose the mates they do? For decades, this question has been approached through the lens of three major hypotheses: good genes, runaway sexual selection, and sensory bias. These hypotheses have had major influences on how we think about sexual selection, but they also fall short in some fundamental ways. First, they each rely on the starting assumption that female mating preferences have a strong heritable basis. But with a few notable exceptions, this has found little support, and rare tests outside of Insecta report low and non-significant preference heritability. Second, there's the Leck paradox. If mating preferences are directional and consistent over time, they should erode genetic variation in preferred traits, and male traits should no longer vary, but that's not what we see. These classic hypotheses also do a poor job of explaining some common empirical patterns, such as preferences that differ among populations of the same species, within populations over time, or among females in the same population. Or there's the fact that many researchers test for an influence of male traits on success and find no relationship at all, even when they're the sort of traits that seem like they should be obviously important. The red of male cardinals does not predict success, even though it seems like it should. All of this suggests that we're missing something in our larger framing of how sexual selection works. Today, I'll explain an idea developed and modeled in collaboration with Courtney Fitzpatrick, Liz Hobson, and Maria Servedio. We propose that females acquire mating preferences by observing others' choices and use context-dependent information to infer which traits are attractive. For example, imagine a population of your favorite taxon, where available males have variable phenotype. A choosing female chooses to mate with male 3, and an observer witnesses this interaction and learns that male 3's phenotype is attractive. The chooser based her choice on the fact that the red of male 3's plumage is slightly more saturated than that of the others, but he's not really that different from male 2 or male 5 in that respect. Instead, to the observer, male 3 stands out for his relatively long tail. The observer learns that long tails are attractive and applies this new information to her future mating choices. Basically, females learn from each other's choices, but they're not mind readers. Instead, they use what makes a male distinctive to infer what it was that made him attractive. The idea of inferred attractiveness is based on three well-supported assumptions. First, that social learning influences mate choice. And indeed, two recent meta-analyses of mate choice copying report a significant overall effect of social information on choice. Females are on average around 2.7 times more likely to pick a male they perceive as being chosen by others. Inferred attractiveness also assumes that observers are learning generalized preferences rather than a preference for a specific individual, and this is supported by direct tests and more meta-analytical support. And finally, the hypothesis assumes that observers learn what makes a male distinctive, which is in line with findings about category learning from the field of cognitive psychology, as well as species that show rare male mating advantages. Support for these three assumptions suggests that inferred attractiveness will affect mate choice. So what patterns would this process produce? We modeled a simplified scenario where males had two traits, A and B. For example, you could think of them as shape and color. And for each trait, there were two options inherited from the father. Shape could be square or circle, and color could be red or blue. Every individual had alleles at both traits. We assume that females live for 3 years and observe others' choices in a population in year 1. Here, this male is chosen by another female. Then females make their own mate choices in years 2 and 3 based on the information they learned in year 1. Observers learn a preference for the trait variant of the chosen male that's rarer in the population. So here, of the traits color and shape, the color of the chosen male is rarest. The observer would learn that color was important and would prefer blue males. If instead this male had been observed as chosen, the observing female would learn that shape was important, since this male has a rare shape but a common color in the population, and she would favor circles in later years. We were interested in evolutionary effects, so we applied the scenario over 100 generations and tracked population-wide changes in female preferences and male allele frequencies. And when we were interested in the effects of context, so we ran the model under different combinations of viability and sexual selection. We varied viability selection from a situation where, for example, red and blue males survived equally well, to a situation where red males were half as likely to survive to adulthood as blue. And we varied sexual selection, which we parameterized as how much more likely a female was to mate with a male that has her preferred trait variant, given that she encounters both. So here, females learn but don't act on whatever preferences they've learned, whereas at this point, females were 10 times more likely to mate with males that have traits they learned to prefer in year 1. So now I'm going to show you the outcomes from 20 replicate runs of the main model to examine the patterns this produces. The plots I'll show summarize female preference extremes, which are when more than 75% of the females in the population showed a preference for the same trait. We chose this as an arbitrary point at which some poor field biologists like me might have a good chance of identifying that pattern from empirical data. More red means more of our 20 model runs showed extremes. And from the same 20 model runs, we also tracked male trait variability. More blue means more runs where variability was maintained for 100 generations. I'll show you the overall pattern and then highlight example runs from situations with high viability selection but low sexual selection, and vice versa. The effects of inferred attractiveness clearly vary across selective contexts. The process produces repeated population-level switches in female preferences over time, particularly when there's strong viability selection. Here, as trait variation is lost, preference in the population, shown by the red line, switches from most females preferring trait B to most preferring A and back again. These blue lines show the frequency of allele 2 for each trait, and you can see that switches in female preference occur as those male traits move toward fixation. In our model, allele 2 had the relative survival benefit, and in situations where viability selection was strong, female preferences favored those adaptive alleles. In contrast, in this area of the plot, where sexual selection was strong and viability selection relatively weak, female preferences changed so frequently that extremes rarely occurred. In this part of the parameter space, these rapidly switching female preferences favored the most costly allele options, which were rarer overall. The implications of this are that certainly effects of inferred attractiveness vary by selective context, but also that these patterns should produce differences over time in which traits are preferred, and by extension, differences among populations, which would likely be out of sync with each other. Because male trait variation is maintained indefinitely under most circumstances, this process offers a plausible resolution to the Leck paradox. And because of differences in which alleles are preferred under different circumstances, this one mechanistic process could in some circumstances produce patterns that look like good genes, a preference for adaptive traits that do influence viability, but look like runaway sexual selection in others. For example, preferences that persist even despite negative fitness consequences. You can get an idea of what to expect in typical situations by focusing on average levels identified in meta-analyses of viability selection or social influence on mate choice. And in this region, we see a lot of variation in female preferences in the population without preference extremes. And male trait variation is maintained. This means that the norm for us folks trying to measure these mating preferences should be differences in preference among females in the same population, and no clear preference even for obvious traits in average circumstances. So what's going on here? The patterns produced by inferred attractiveness are not as simple as either positive or negative frequency dependence. To be the target of female preference, a trait must be common enough to be observed in a chosen male, and yet also rare enough to be unusual in the population so that it stands out as distinctive to the female. In the paper, which is freely available from PLOS Biology, we also explored what happens if viability selection on the two traits is unequal, which should be the norm in natural populations, or if sexual selection was stronger on one trait than the other. This would be analogous in our model to what would happen if females more readily perceived one trait than another. In other words, if they had a sensory bias. We also examined what happens if traits are insensitive to selection. For example, if they're environmentally determined or age-dependent. The overall patterns that result are interestingly different, but the underlying mechanism is the same. Patterns in preference and traits are driven by changes in relative allele frequencies perceived by females. We also compare the patterns produced by inferred attractiveness to those from other types of models to underscore how different this model is. And you can read all about that in the paper linked at the right. I do want to point out that mate choice by inferred attractiveness does not exclude the action of other processes. In particular, sensory bias should matter. While inferred attractiveness proposes a non-genetic preference underlying choice, sensory systems do, of course, have a genetic basis. And this process should apply to any species capable of social learning, so that includes organisms from Drosophila to humans. Finally, it does not require that social learning be selected for in a mate choice context. In fact, we think this behavior would be more likely to result from selection to pay attention to conspecifics as individuals learn about predators, food, or nest sites. Inferred attractiveness is a new hypothesis that offers an explanation why females favor certain traits and simultaneously why there's so frequently no particular trait favored or obvious traits are ignored. This mechanism produces patterns that look like good genes in some circumstances and runaway sexual selection in others, but it is none of the above because preferences are not genetically determined. And finally, inferred attractiveness is a dynamic process that presents a plausible solution to the Leck paradox. Thank you to everyone who contributed to this work, and I would be happy to take questions either in online office hours or by email.